What Earth was really like before animals. Welcome, dear listener, to a journey through the deepest recesses of time, a tale of our planet before the first creature stirred, before eyes beheld the light, before any sound echoed beyond the crash of waves or the rumble of volcanoes. This is the story of Earth in its primordial silence, a world without animals, spanning billions of years where life was but a whisper in the vastness of rock and water. Imagine a globe shrouded in mystery, where the only architects were microbes, and the only dramas were chemical symphonies played out in oceans and atmospheres. We'll drift through this ancient narrative slowly, like sediment settling on the seafloor, uncovering layer by layer what Earth was truly like before the dawn of animal life. Settle in, let your mind wander back, and allow the calm of deep time to envelop you. Our story begins not with life, but with the birth of the planet itself. Some 4.6 billion years ago, in the swirling chaos of the early solar system, Earth coalesced from a disk of dust and gas orbiting a young sun. Picture it, a molten ball of rock bombarded by asteroids and comets, its surface a sea of lava glowing under a sky thick with debris. This was the Hadean Aeon, named after Hades, the Greek underworld, for it was a hellish time of unrelenting heat and violence. Temperatures soared to thousands of degrees, melting any nascent crust into a global magma ocean. Volcanic eruptions spewed gases, carbon dioxide, water vapor, nitrogen, and traces of methane, forming the first tenuous atmosphere. But this air was nothing like today's. It was heavy, toxic, and devoid of free oxygen, a suffocating blanket that trapped heat and fueled storms of unimaginable fury. As the bombardments waned, around 4.4 billion years ago, Earth's surface began to cool. The magma ocean solidified into a thin crust of basalt, cracked and reforming endlessly under the strain of internal heat. Water, delivered by icy comets or outgassed from the mantle, condensed into the first oceans. These were not the blue expanses we know. They were acidic, mineral-laden soups, boiling in places where hydrothermal vents pierced the seafloor. The Moon, formed from a cataclysmic collision with a Mars-sized body called Theia, tugged at these nascent seas, creating tides that sloshed against barren shores. Day and night cycled rapidly. Earth spun faster then, with days perhaps as short as 6 hours, and the sun was dimmer, a faint star casting feeble light on a world still finding its footing. In this barren cradle, the stage was set for life's emergence. But before we delve into that miracle, let's linger on the geology that shaped it. The Hadean gave way to the Archean Aeon around 4 billion years ago, as the crust stabilized into the first continents, tiny proto-landmasses of granite floating on denser mantle rock. Plate tectonics, that great recycler of Earth's surface, kicked into gear, driven by convection currents in the molten interior. Mountains rose and eroded, volcanoes built islands, and rifts tore open to spill fresh lava. The atmosphere evolved too. Photodissociation by ultraviolet rays split water molecules, releasing hydrogen that escaped to space and leaving oxygen to react with iron in the rocks, rusting the planet's surface in bands of red. Yet amid this tumult, the first hints of habitability emerged. The oceans cooled to temperatures where liquid water could persist, and dissolved minerals provided the building blocks for complexity. Zircon crystals from this era, tiny time capsules found in Western Australia, tell us of liquid water as early as 4.4 billion years ago, astonishingly soon after Earth's formation. These grains, etched with isotopic signatures, reveal a world already cycling carbon and nurturing the potential for life. No animals roamed, of course. The land was sterile, the seas empty of anything but dissolved gases and salts. But in the quiet depths, near those hydrothermal vents, black smokers belching superheated water rich in hydrogen sulfide and metals, chemical reactions brewed the precursors of biology. Let's pause to envision one such vent field, perhaps on the floor of a shallow Archean sea. Jets of hot fluid mingle with cold seawater, precipitating minerals into towering chimneys. Here, in the gradient between heat and cold, energy flows freely. Hydrogen from the vents reacts with carbon dioxide to form methane, and simple organic molecules, amino acids, sugars, lipids, assemble in the porous rock. This is the warm little pond Darwin imagined, but deeper and more dynamic. Theories abound: metabolism first, where energy cycles precede genetics, or RNA world, where self-replicating molecules bridge chemistry and life. Whatever the spark, by 3.8 billion years ago, evidence suggests life had begun: simple prokaryotes, bacteria-like cells thriving in extreme conditions. These earliest life forms were anaerobes, feasting on chemical energy without oxygen. They clustered in mats on the seafloor, layering sediment with their waste, forming stromatolites, dome-shaped structures that are among the oldest fossils we have. In places like Shark Bay, Australia, modern analogues persist, but imagine the Archean versions blanketing vast shallows under a hazy orange sky. The sun's UV rays pierce the thin ozone layer, mutating early genomes but also driving evolution. Cyanobacteria, those pioneering photosynthesizers, appeared around 3.5 billion years ago, harnessing sunlight to split water and release oxygen as a byproduct. This was revolutionary but slow. For eons, that oxygen bound to iron in the oceans, sinking as rustiform banded iron formations, vast deposits that today supply our steel. As we drift through this narrative, consider the silence. No birdsong, no insect buzz, no footsteps on the shore. The soundtrack is elemental: wind whipping over lava fields, rain pattering on cooling rock, waves lapping at empty beaches. Earth's magnetic field, generated by the churning core, shielded the atmosphere from solar winds, preserving this fragile setup. Comets still occasionally struck, delivering water and organics, while massive volcanic events like the formation of large igneous provinces reshaped continents. One such event perhaps flooded the surface with basalt, creating plateaus that weathered into fertile soils, though no plants yet existed to claim them. Deeper into the Archean, around 3 billion years ago, the first supercontinent may have formed: Volbara or masses of land colliding as plates danced. Climate swung wildly. Greenhouse gases kept things warm, but without oxygen's ozone shield, the surface was harsh. Life, confined to water, diversified: methanogens producing methane in anoxic muds, sulfur bacteria cycling elements in vents. Genetic evidence from modern microbes traces back to a last universal common ancestor, LUCA, a simple cell with a genome of perhaps 500 genes, living in hot, alkaline environments. From LUCA branched bacteria and archaea, the domains that dominated for billions of years. But let's not rush, this is a story of patience. The Archean seas teemed with invisible life, but the land remained a desert of rock. Erosion carved canyons, rivers of acidic water dissolved minerals, and lightning storms fixed nitrogen into forms life could use. The moon loomed larger, its craters fresh from impacts, and tides were fiercer, perhaps influencing early biochemical cycles. Asteroid strikes, though less frequent, could vaporize oceans temporarily, stressing life but selecting for resilience. In the rock record, we find graphitic carbon, remains of those early cells, interwoven with minerals, a testament to life's tenacity. As the eon progressed, oxygen levels ticked up slightly, oxidizing the surface and creating new niches. Red beds appeared, iron-rich soils staining the landscape. Microbial mats evolved strategies. Some burrowed into sediment, others formed biofilms that trapped particles. In shallow lagoons, evaporation concentrated salts, forming evaporites that preserved delicate structures. The atmosphere stabilized with about 1% oxygen, enough to change chemistry but not yet transform the world. This was the calm before the storm, a prelude to the Great Oxidation Event that would redefine Earth. Imagine floating above this ancient world, continents adrift on a blue marble, volcanoes punctuating the horizon, storms brewing over endless seas. No green mantle covers the land, it's gray and black, pocked with craters. Below, in the depths, life pulses quietly, a hidden engine driving planetary change. This is Earth before animals, a realm of rock, water, and microbes where time unfolds in geological size. We'll pick up from here in the next part, diving deeper into the rise of oxygen and the transformations it wrought. For now, let this image linger, a silent planet pregnant with possibility, waiting for the next chapter in its epic saga. A time long before the first animal took a breath or left a footprint. This is the story of the Precambrian era, encompassing more than 88% of our planet's 4.6 billion year timeline, a vast expanse where life was simple, the land barren, and the oceans a cradle of chemical wonders. We'll unfold this narrative like layers of sediment, building a picture of a world shaped by geology, chemistry, and the slow emergence of biology. No drama of predators and prey, no forests or reefs, just the quiet rhythm of a planet in its youth. Let's begin at the beginning with the birth of Earth, and let the calm of deep time wash over you as we journey through this forgotten world. The story opens in the chaos of the early solar system, about 4.6 billion years ago. The sun had just ignited from a collapsing cloud of gas and dust, and the remaining material formed a spinning disk where planets coalesced. Earth formed through accretion, tiny particles clumping into larger bodies over millions of years. By the time it reached planetary size, the heat from collisions and radioactive decay melted it into a magma ocean, a global sea of molten rock hundreds of kilometers deep. The surface glowed with intense heat, temperatures soaring to 2,000–3,000 degrees Celsius, and heavier elements like iron sank to form the core, while lighter silicates rose to the mantle. This differentiation released more heat, keeping the interior molten. The young Earth was a hellish place, the Hadean Aeon, named for its infernal conditions. Bombardments from asteroids and comets were relentless, each impact creating craters and ejecting debris. The atmosphere emerged from volcanic outgassing, gases trapped in the mantle released as lava cooled. Water vapor, carbon dioxide CO2, nitrogen N2, ammonia NH3, methane CH4, hydrogen sulfide H2S, and hydrogen H2 formed a thick, toxic envelope. This air was reducing, lacking free oxygen O2, and it acted as a greenhouse, trapping heat to counter the faint young sun, which was 25–30% dimmer than today. Without it, Earth might have frozen, but instead the surface remained hot and stormy. Lightning streaked through the clouds, UV radiation from the sun penetrated deeply, and the planet spun fast, with days as short as 5–6 hours. The moon's birth added to the drama. A Mars-sized body, Theia, collided with Earth around 4.5 billion years ago, splashing material into orbit that coalesced into our satellite. This event tilted Earth's axis, creating seasons, and the moon's gravity slowed the planet's spin over time, while stabilizing its tilt to prevent extreme climate shifts. As the bombardments tapered off around 4 billion years ago, the surface cooled. The magma ocean solidified into a thin crust of basalt, cracked by tectonic activity. Water, delivered by icy comets or outgassed from the interior, condensed into oceans. Evidence from ancient zircons in Australia's Jack Hills, crystals dating to 4.4 billion years ago, shows oxygen isotopes indicating liquid water, with some grains even suggesting continental crust early on. These oceans were vast, covering most of the planet, and they were hot, acidic, and mineral-rich. The pH was low from CO2 dissolving to form carbonic acid, and dissolved iron, silica, and other metals made the water a chemical soup. Hydrothermal vents on the seafloor, black smokers spewing superheated fluid at 350–400 degrees Celsius, created dynamic environments where seawater circulated through hot rock, leaching minerals and creating pH and temperature gradients. This is where life likely began, between 4.2 and 3.8 billion years ago. The origin is a puzzle, but theories point to vents or shallow pools. In alkaline vents, serpentinization produced hydrogen, which reacted with CO2 to form methane and organics. Gradients drove proto-metabolism, with minerals catalyzing reactions. The RNA world hypothesis suggests self-replicating RNA molecules bridged chemistry and biology, enclosed in lipid membranes to form proto-cells. The first life was simple, prokaryotes like bacteria and archaea, anaerobic, using chemical energy from hydrogen or sulfur. They reproduced by division, swapped genes horizontally, and adapted to extremes. The Archean Eon from 4 to 2.5 billion years ago saw the crust thicken into stable cratons. Plate tectonics started with subduction and spreading, building volcanic islands and proto-continents. Cometate lavas hotter than modern basalts flowed, indicating a hotter mantle. The atmosphere stabilized with N2 dominant, CO2 high for warmth, and methane from life or vents. The magnetic field generated by the core's dynamo protected the air from solar erosion. Life flourished in water, forming mats that built stromatolites, layered structures from cyanobacteria trapping sediment. The oldest, at 3.5 billion years in Pilbara, Australia, show microbial communities. Cyanobacteria invented oxygenic photosynthesis, splitting water to produce O2. But oxygen was scarce, oxidizing iron to form banded iron formations, BIFs, like those in Minnesota's Iron Range. The ocean was ferruginous, with green iron-rich waters. The land was empty, eroded by rain and wind, forming red soils from oxidized iron. Climate was warm, 40–60 degrees Celsius, with occasional glaciations. Supercontinents like Valbara formed, colliding cratons. Life was resilient, surviving impacts and volcanic events. As oxygen levels rose slowly, the stage was set for the Great Oxidation Event, but that comes next. Let the Archean serenity linger, a world of rock, water, and microbes waiting for change. The narration continues from the cusp of the Great Oxidation Event in the next part. Control 12 to get a more accurate and detailed narration. I should research the topic further to ensure the content is comprehensive and up to date. The video is a YouTube live, so using browse page to get the transcript or key points would help expand to the required length. Function call for browsing the YouTube page to extract the full description, comments, or any transcript if available, but YouTube pages don't have full transcripts. However, the instructions can be to summarize the content or key points from the video description and related info. Since it's a live video, perhaps use web search for transcript of what Earth was really like before animals, the snoozy scientist, or similar to find if there's a transcript. To make a long narration, I can use web search for Earth before animals Precambrian era detailed history to get sources. Since the user wants a narration on the topic, and to make it 30–40K words but divided, I can plan to generate based on knowledge, but to substantiate, search for distribution of sources. The topic is not controversial, but to be thorough. Let's use web search to get key facts and structure for the narration. Query detailed history of Earth before animals Precambrian Eon timeline events, num results 20 to get good coverage. Control 12 what Earth was really like before animals let us embark on a tranquil voyage through the annals of deep time, a narrative woven from the silent testimonies of rocks, isotopes, and ancient whispers preserved in the Earth's crust. This is the chronicle of our planet before the advent of animals, a realm spanning over 4 billion years, where the drama unfolded not in the scurry of creatures or the rustle of leaves, but in the slow symphony of geological forces, chemical reactions, and the subtle emergence of life in its most primordial forms. Picture a world unadorned by forests or fauna, where oceans churned under hazy skies, volcanoes sculpted landscapes, and microscopic entities laid the groundwork for all that would follow. In this extended narration, we'll traverse the Precambrian eons, the Hadean, Archean, and Proterozoic, unveiling a Earth that was raw, resilient, and profoundly alien. We'll proceed deliberately, allowing each epoch to unfold like sediment settling on an ancient seafloor, evoking the calm wonder of a cosmos patiently crafting its masterpiece. As we begin, imagine yourself adrift in the void, witnessing the birth of a world from cosmic dust. Our tale commences with the genesis of Earth itself, approximately 4.6 billion years ago, amid the swirling remnants of a stellar nursery. The solar system coalesced from a vast cloud of gas and dust, the sun igniting at its heart while the surrounding material flattened into a protoplanetary disk. Within this disk, particles of ice, rock, and metal collided and aggregated, forming planetesimals that grew into planets through a process of accretion. Earth, one such world, accumulated mass rapidly, its interior heating from gravitational compression and radioactive decay until it became a fully molten sphere, a glowing orb of magma suspended in the cold expanse of space. The surface was a seething ocean of liquid rock, temperatures exceeding 2,000 degrees Celsius, with no solid ground to speak of. Heavy elements like iron and nickel sank to form the planet's dense core, while lighter silicates rose to create the mantle, releasing tremendous energy in the process. This differentiation not only structured Earth's layers but also generated the initial heat that still warms our world today. This earliest chapter, known as the Hadean Eon, stretched from about 4.54 billion to 4 billion years ago, a period aptly named after Hades, the Greek underworld, for its infernal chaos. The young Earth endured relentless bombardments from asteroids, comets, and planetesimals left over from the solar system's formation. These impacts were cataclysmic, each one vaporizing vast swaths of the surface and ejecting molten debris into space. One monumental collision around 4.5 billion years ago involved a Mars-sized body dubbed Theia, which struck Earth with such force that it blasted a portion of the mantle into orbit. This ejected material coalesced to form the Moon, an event that not only gave us our celestial companion but also tilted Earth's axis, introducing seasons, and slowed its rotation from initial days of mere hours to something closer to our modern cycle. The Moon's gravity would later stabilize Earth's tilt, preventing wild climatic swings, and its pull created powerful tides that stirred primordial seas. Amid this violence, the first atmosphere emerged, born from volcanic outgassing as gases trapped in the molten interior escaped. Water vapor, carbon dioxide, nitrogen, ammonia, methane, and hydrogen sulfide formed a thick, hazy envelope that trapped heat like a blanket, countering the faint light of the young sun, which shone with only about 70% of its current brightness. This greenhouse effect kept the surface warm enough to remain molten, but as the planet cooled and the bombardments waned, water vapor condensed into rain, pooling to form the first oceans around 4.4 billion years ago. Evidence for this comes from ancient zircon crystals in Western Australia, tiny minerals that preserve isotopic signatures indicating the presence of liquid water and even hints of continental crust far earlier than once thought. These oceans were not the serene blue expanses we know. They were hot, acidic broths with pH levels around 6, laden with dissolved minerals from volcanic activity and comet deliveries. The seafloor was riddled with hydrothermal vents, black smokers spewing superheated, mineral-rich fluids, creating dynamic chemical gradients where heat met cold seawater. The Hadean landscape, if one could call it that, was a barren hellscape of lava flows, impact craters, and fleeting crusts that cracked and reformed under tectonic stresses. Plate tectonics may have begun in a primitive form, driven by convection in the mantle, but the surface was too hot and unstable for lasting continents. The sky was a perpetual twilight, choked with dust and volcanic ash, pierced by lightning storms that fixed nitrogen and drove early chemical reactions. Ultraviolet radiation from the sun bombarded the unprotected surface, breaking molecular bonds and fostering the synthesis of organic compounds. Comets, those icy wanderers from the outer solar system, continued to pelt Earth, delivering water, carbon, and perhaps the seeds of life in the form of amino acids and other organics. Life's origins likely stirred in this tumultuous cradle, though the Hadean left scant direct evidence. No rocks survived from this eon, erased by the constant resurfacing. Theories suggest that in the alkaline environments of hydrothermal vents, serpentinization reactions produced hydrogen gas, which combined with carbon dioxide to form methane and simple organic molecules. H gradients acted like natural batteries, energizing proto-metabolic cycles. Alternatively, in shallow ponds or tidal pools, lightning and UV light sparked the formation of amino acids, as demonstrated by the famous Miller-Urey experiment in the 1950s. Lipids self-assembled into membranes, RNA molecules replicated, bridging the gap between chemistry and biology in what scientists call the RNA world. By the close of the Hadean, around 4 billion years ago, the planet had cooled sufficiently for a more stable crust to form, marking the transition to the Archean Eon. The late heavy bombardment, a final intense barrage of impacts peaking around 3.9 billion years ago, may have been triggered by orbital shifts in the outer planets, reshaping the surface one last time. Envision this Hadean Earth, a molten marble adrift in space, its surface a canvas of fire and shadow, oceans steaming under a greenhouse sky. No life yet animated its waters, but the building blocks were assembling, the stage set for the microbial revolution to come. The planet's core churned, generating a magnetic field that shielded the atmosphere from solar winds, preserving the fragile conditions for future habitability. Volcanoes built ephemeral islands, rains eroded nascent rocks, and the moon loomed large, its craters fresh testaments to shared bombardments. This was a world of extremes where destruction birthed creation, and time measured in eons allowed chaos to yield order. As the Hadean faded, the Archean dawned, bringing the first robust rock record and the undeniable spark of life. But that is where we shall resume in the next segment of our narration, delving into the formation of continents, the rise of microbes, and the quiet transformation of a barren world. In this opening part, we've laid the foundation, much like the cooling crust itself, preparing for the layers to come. Let the imagery of this primordial Earth linger in your mind, a silent, fiery genesis, patient and profound. We now step forward from the molten chaos of the Hadean into the Archean Eon, the second great chapter in Earth's deep time biography. The transition around 4 billion years ago was not abrupt. It was a gradual cooling, a settling of the crust, a quieting of the most violent bombardments. The planet had exhaled its first fiery breath and now began to breathe more steadily. The Archean spans from roughly 4 to 2.5 billion years ago, an immense stretch during which the foundations of continents appeared, the first unambiguous signs of life emerged, and the microbial world quietly took command of the planet's chemistry. No animals, no plants, no visible ecosystems on land, only water, rock, and the invisible labor of single-celled organisms reshaping a world that had previously known only physics and chemistry. By the beginning of the Archean, the surface had cooled enough for a permanent, though thin and fragile, basaltic crust to persist. Small proto-continents, granitic masses lighter than the surrounding mafic ocean floor, began to form through partial melting and differentiation in subduction-like zones or above mantle plumes. These earliest pieces of continental crust, preserved today as ancient cratons, the Pilbara in Western Australia, the Kopval in southern Africa, the Superior in North America, the Slave in Canada, are the oldest surviving pieces of Earth's outer shell. They were small islands or microcontinents at first, scattered across a globe that was still overwhelmingly ocean-covered, perhaps 80–90% water surface. Plate tectonics, though perhaps operating differently than today, hotter mantle, thinner plates, faster spreading, was already active. Subduction zones recycled crust, arc volcanism built new land and rifting opened basins. Large igneous provinces, massive outpourings of basalt, flooded regions hundreds of thousands of square kilometers in extent. The mantle was hotter than now, producing cometates, very high temperature, low viscosity lavas that flowed rapidly across the surface like liquid fire, forming thick, magnesium-rich layers now altered into distinctive greenstone belts. The atmosphere remained anoxic, no free oxygen to rust iron or support aerobic respiration. Nitrogen likely dominated ~80%. Carbon dioxide was still very high, hundreds to thousands of times modern levels. Methane and other trace greenhouse gases were abundant. This thick greenhouse blanket compensated for the faint young sun paradox. Despite receiving only ~75–80% of today's solar energy, Earth avoided becoming a global snowball because of the powerful heat-trapping effect of CO2, CH4, and possibly ethane or carbonyl sulfide. Average surface temperatures may have ranged from 55–85 degrees C in many models, though some evidence suggests episodes cooler than that. The sky would have appeared orange-brown or deep amber under perpetual haze. Rayleigh scattering was less effective without much oxygen or ozone, so shorter wavelengths were not scattered away as efficiently. Oceans covered most of the planet and were chemically very different from today. They were rich in dissolved ferrous iron, Fe2, making them a pale green or murky olive color in shallow areas. Silica concentrations were high because there was no biological removal mechanism. Diatoms and sponges had not yet evolved. The pH was mildly acidic to near neutral. Hydrothermal circulation at mid-ocean ridges and in back-arc basins was vigorous, exchanging heat and chemicals between crust and ocean on a massive scale. It is in this setting that we find the first compelling evidence for life. The oldest widely accepted morphological evidence of life appears around 3.43 billion years ago in the Dresser Formation and Strelley Pool Chert of the Pilbara Craton, Western Australia. These rocks preserve conical, demal, and stratiform stromatolites, layered sedimentary structures formed by microbial mats trapping and binding sediment grains while precipitating minerals, mainly carbonate. Modern analogues exist in Shark Bay, Western Australia, and in hypersaline lagoons elsewhere, built by cyanobacteria. In the Archean, similar communities likely thrived in shallow, sunlit waters where photosynthesis could occur. Around the same time, or slightly earlier, 3.5–3.7 Ga, carbon isotope signatures in graphitic material from Greenland Isua supercrustal belt and Labrador show strong enrichment in 12C relative to 13C, a fractionation pattern almost certainly produced by biological processes. Enzymes preferentially use the lighter isotope. While debated, these signatures are consistent with microbial carbon fixation via pathways such as the Calvin cycle or Wood-Lundahl pathway. The organisms responsible were prokaryotes, bacteria and archaea. No eukaryotes yet existed. The last universal common ancestor, LUCA, likely lived sometime between 4.2 and 3.8 billion years ago in a hot, chemically rich environment, perhaps near alkaline hydrothermal vents. From LUCA diverged the two primary prokaryotic domains. Early metabolisms were anaerobic, methanogenesis producing CH4 from CO2 and H2, acetogenesis, fermentation, sulfur reduction, iron reduction. Anexogenic photosynthesis using bacteria chlorophyll appeared first, utilizing hydrogen sulfide, ferrous iron, or organic compounds as electron donors instead of water. Then, sometime between and billion years ago, oxygenic photosynthesis evolved in the cyanobacterial lineage. This was the single most important metabolic innovation in Earth history. For the first time, organisms used water H2O as an electron donor, releasing molecular oxygen, O2, as a waste product. 2H2O plus light right arrow 4H plus 4E dash plus O2 oxygen was initially poisonous to most early life forms. It reacted rapidly with ferrous iron dissolved in the oceans, precipitating ferric iron Fe3 as insoluble oxides and hydroxides, hematite Fe2O3, magnetite Fe3O4, and greenolite. These iron minerals sank to the seafloor, forming the famous banded iron formations, BIFs, that characterize much of the late Archean and early Proterozoic rock record. BIFs are rhythmic layers of iron-rich chert and iron oxide, often deposited in deep water settings far from shore. They represent the planet's way of mopping up the first photosynthetic oxygen, preventing it from accumulating in the atmosphere for an extraordinarily long time. Microbial mats, layered communities of different metabolic types, became widespread in shallow waters. Phototrophs lived at the top using sunlight. Beneath them, chemotrophs used sulfide, iron, or organic matter produced above. These mats trapped sediment, precipitated carbonate or silica, and created the stromatolite record that persists for billions of years. On land there was still nothing, no soils, no microbial crusts thick enough to leave clear fossils, no evidence of subaerial life until much later. The continents were small, low relief, frequently inundated. Weathering was intense under a CO2-rich atmosphere and frequent acid rain. Rivers carried dissolved silica and iron to the oceans, contributing to chert and iron deposits. Asteroid and comet impacts continued, though less frequently than in the Hadean. Large igneous events flooded regions with basalt. Supercontinent cycles may have begun. Some reconstructions place the first true supercontinent, Valbara or around 3.63 billion years ago, though evidence is fragmentary. Throughout the Archean, life was confined to water. The biosphere was microbial, metabolically diverse, genetically fluid, horizontal gene transfer was rampant, and extraordinarily resilient. Extinction events occurred, perhaps acts or massive volcanic outpourings, but life persisted and diversified. By the end of the Archean (2, 5 billion years ago, something profound was changing. Oxygen production had slowly outpaced the ocean's capacity to buffer it through iron precipitation. Ferruginous oceans began to give way to euxinic, sulfide-rich, and eventually, oxic conditions in some places. Atmospheric oxygen started to rise, marking the onset of the Great Oxidation Event, GOE, one of the most dramatic turning points in Earth history. We now cross one of the most decisive thresholds in Earth's entire history, the moment when oxygen, that quiet poison produced by ancient microbes, finally began to accumulate in significant quantities in the oceans and atmosphere. This turning point is known as the Great Oxidation Event, or GOE, and it fundamentally altered the chemistry of the planet, reshaped the biosphere, and set the long, slow stage for everything that would eventually follow, including, billions of years later, complex multicellular life in animals. The Great Oxidation Event is not a single, sharp moment. It unfolded gradually between approximately 2.45 and 2.32 billion years ago, with important precursor activity stretching back perhaps several hundred million years earlier, and important after effects continuing for another billion years or more. Geologists mark its onset by the disappearance of certain mineral signatures and the appearance of others in the rock record. For roughly the first 300–500 million years of oxygenic photosynthesis, roughly 32.7 to 4 billion years ago, almost all the oxygen produced by cyanobacteria was immediately consumed. The primary sink was dissolved ferrous iron, Fe2, in the oceans. As soon as O2 appeared, it reacted with Fe2 to form ferric iron, Fe3, which is far less soluble and precipitated out as iron oxides, hematite, magnetite, goethite, creating the massive banded iron formations we discussed earlier. These BIFs are among the most characteristic rocks of the late Archean and early Proterozoic. They required anoxic deep oceans to keep iron dissolved and transportable, but at least locally oxygenated surface waters where photosynthesis occurred. By around 2.45 billion years ago, however, the supply of dissolved iron began to be exhausted in many parts of the ocean. The oxygen buffer was becoming saturated. Once the ferrous iron was largely precipitated out, new oxygen molecules had fewer easy reaction partners. Some reacted with methane and other reduced gases in the atmosphere, but eventually, slowly, inexorably, free O2 started to build up in the surface ocean, and for the first time in Earth history, began to leak into the atmosphere in meaningful quantities. The most diagnostic geological evidence for the GOE comes from several independent lines. Mass-independent fractionation of sulfur isotopes, MIFS, before the GOE. Ultraviolet radiation could penetrate deep into the atmosphere because there was no ozone layer. UV light split SO2 molecules in unusual ways, producing sulfur isotopes 33S and 36S that do not follow normal mass-dependent fractionation patterns. These anomalous signatures are preserved in Archean sedimentary rocks, pyrite, barite, etc. After the GOE, once an ozone shield formed, UV was blocked higher in the atmosphere, and this MIFS signal disappears almost completely from the rock record around 2.3 to 2.4 Ga. The disappearance of MIFS is one of the sharpest and most globally synchronous markers of atmospheric oxygenation. Red beds, terrestrial sedimentary rocks older than Ga are typically gray or green, reduced iron. After the GOE, red beds, sandstones, and shales stained red by ferric iron oxides become common for the first time. Oxygen in rain and groundwater oxidized iron minerals at the surface. Cessation of major BIF deposition. The peak of BIF formation ends abruptly around 2.4 Ga in most regions, although smaller, later BIFs exist. With less dissolved iron available, oxygen could no longer be so efficiently buried. Manganese deposits around the same time. Large sedimentary manganese deposits appear, e.g., the Kalahari manganese field in South Africa. Manganese requires stronger oxidizing conditions than iron to precipitate, suggesting that surface waters became more oxidizing. The rise of atmospheric oxygen during the GOE is estimated to have been modest by modern standards, perhaps from near 0 to 0.11% of present atmospheric levels, PAL, possibly even lower in some models. Yet even this small amount was revolutionary. Oxygen is a powerful oxidant. Its accumulation was catastrophic for most of the existing microbial world. Strict anaerobes that had dominated for billions of years suddenly faced a toxic gas. Many lineages went extinct or retreated to oxygen minimum zones, deep sediments, hydrothermal vents, and other refugia. This event is sometimes informally called the oxygen catastrophe or oxygen crisis from the perspective of anaerobic life. At the same time, oxygen opened entirely new ecological opportunities. Aerobic respiration became possible. Using O2 as the terminal electron acceptor yields far more energy per molecule of organic carbon oxidized than any anaerobic pathway, 18 times more ATP than fermentation. Organisms capable of aerobic metabolism gained a huge competitive advantage in environments where oxygen was available. The ozone layer began to form. Even at low atmospheric O2 levels, enough O3 was produced to absorb most incoming UVC and much UVB radiation. This reduced the flux of harmful ultraviolet light reaching the surface, making shallow water and eventually terrestrial habitats more habitable. Oxidative weathering of continental rocks accelerated. Pyrite and other sulfide minerals exposed at the surface oxidized, releasing sulfate to rivers and oceans and increasing seawater sulfate concentrations dramatically, from 1 mm to perhaps 5-15 mm. The GOE also coincided with the possible first Snowball Earth glaciations, although the major Huronian glaciations, 2.4-2.1 Ga, are debated in timing and extent. Some researchers link them to the drawdown of greenhouse gases, especially methane, as oxygen reacted with CH4, weakening the greenhouse effect and allowing ice to advance. Others argue the glaciations were caused by tectonic factors or changes in continental weathering. Regardless, the early Proterozoic saw at least 2 and possibly 3 major glacial intervals during which ice may have reached low latitudes. After the GOE, Earth did not emit oxygen-rich world we know today. The deep oceans remained anoxic or euxinic, sulfide-rich for another billion years or more. Atmospheric oxygen likely fluctuated, with possible temporary declines. Major pulses of oxygenation occurred much later. Yet the GOE was irreversible in one crucial sense. It fundamentally changed the redox state of the surface environment. The planet crossed a geochemical Rubicon. Once oxygen became a persistent component of air and surface water, evolutionary pathways were permanently redirected toward organisms that could tolerate or even exploit oxygen. Microbial communities adapted. Cyanobacteria continued to produce oxygen, but now some of it escaped burial. Aerobic heterotrophs proliferated. sulfur-cycling microbes shifted towards sulfate reduction in euxinic settings. Iron-oxidizing bacteria formed microbial mats in oxygenated surface waters. Stromatolites reached their greatest diversity and abundance during the early Proterozoic, suggesting that shallow water microbial ecosystems thrived under the new, partially oxygenated conditions. The land surface remained biologically barren, no soils, no microbial mats thick enough to leave widespread evidence, no plants or animals. Weathering produced thick regoliths, and rivers delivered increasing amounts of sulfate, molybdenum, and other elements to the oceans. By 1.8 billion years ago, another major transition was underway, the first widespread appearance of eukaryotic cells. That eukaryotic revolution, the slow oxygenation that followed the GOE, the return of global glaciations, and the strange, quiet billion-year interval sometimes called the boring billion, will form the core of our next chapter. We emerge now from the dramatic chemical upheaval of the Great Oxidation Event into a longer, quieter, more enigmatic chapter of Earth's history, the Middle Proterozoic, roughly 2 to 1 billion years ago. This interval is sometimes nicknamed the boring billion, not because nothing happened, but because on the surface, very little seems to have changed dramatically for an extraordinarily long stretch of time. Oxygen levels rose only slowly after the initial GOE pulse. The deep oceans remained largely anoxic or euxinic, sulfide-rich. Stromatolites continued to dominate shallow seafloors, and no complex multicellular organisms or animals appeared. Yet beneath this apparent stability, several of the most important biological transitions in Earth history quietly took place. By tilde 85 1.6 billion years ago, roughly 600 million years after the main phase of the GOE, the first widely accepted eukaryotic fossils appear in the rock record. These are not yet complex algae or proto-animals, but simple, single-celled eukaryotes with nuclei and, in many cases, other membrane-bound organelles. The eukaryotic cell is one of the most profound innovations in the history of life. Unlike prokaryotes, eukaryotic cells possess a true nucleus enclosed by a double-membrane mitochondria. In almost all lineages, in many lineages, other organelles, plastids, endoplasmic reticulum, Golgi apparatus, etc., a dynamic cytoskeleton allowing phagocytosis, cell crawling, and complex internal organization, linear chromosomes with multiple linear pieces of DNA and sophisticated mitotic machinery, the lading. And now overwhelmingly supported explanation for the origin of eukaryotes is the endosymbiotic theory, most comprehensively developed by Lynn Margulies in the 1960s-80s, and now backed by genomic, phylogenetic, and ultrastructural evidence. The key event was an endosymbiosis between an archaeal host cell and an alpha protobacterium that was engulfed but not digested. Over evolutionary time, this bacterium became the mitochondrion, providing the host with vastly more efficient aerobic respiration. Genomic studies show that the closest living relatives of mitochondria are alpha protobacteria, e.g., rickettsials, and that the eukaryotic nuclear genome contains hundreds to thousands of genes of clear alpha protobacterial origin. A second major endosymbiosis occurred later, probably between tilde 1.6 and tilde 1.2 billion years ago, when a eukaryotic lineage that already possessed mitochondria engulfed a cyanobacterium. This endosymbiont was retained and eventually became the plastid chloroplast, found in plants, green algae, red algae, glaucophytes, and their descendants. Again, genomic evidence is decisive. Plastid genomes are clearly cyanobacterial in origin, and many hundreds of cyanobacterial genes have been transferred to the host nucleus. The appearance of the first eukaryotic cells therefore required oxygen, both because mitochondria use O2 to generate ATP efficiently, and because the initial host cell likely already possessed some capacity to tolerate or manage low levels of oxygen. The GOE and subsequent slow oxygenation created the redox conditions that made aerobic eukaryotes metabolically competitive. The oldest widely accepted eukaryotic body fossils are large, up to ~100 μm, organic walled microfossils with complex wall ornamentation, e.g. Valeria laphostriata, Schuiauspheridium macroreticulatum, Tapania plana, found in ~1, 65 1.45 billion-year-old rocks in Australia, China, and Siberia. Some show evidence of binary division, others possible existent structures or processes that suggest sexual reproduction or resting stages. Around 1.21 billion years ago, the fossil record becomes more diverse. Bangiomorpha pubescens, Ga, Arctic Canada, is widely regarded as the oldest fossil red alga. It shows clear multicellular organization with differentiated holdfast cells, uniseriate filaments, and reproductive structures, sporangia. This implies that sexual reproduction, alternation of generations, had already evolved by this time, a prerequisite for complex multicellularity in many lineages. Other fossils from ~1, 0.8 Ga e.g., Arisphera geraldi from the Arctic, red algal filaments, possible green algal remains, demonstrate that simple multicellular and even tissue-grade organization had appeared in several eukaryotic lineages long before animals. Yet despite these advances, the Proterozoic biosphere remained overwhelmingly microbial. Stromatolites reached their peak diversity and abundance between, until ~1.0 billion years ago, then began a long decline, possibly due to the appearance of eukaryotic grazers that disrupted mat communities. The deep oceans stayed anoxic or euxinic. Euxinic conditions were widespread on continental shelves. Atmospheric oxygen is estimated to have remained low, perhaps 110% PAL during much of the boring billion, enough to support aerobic respiration and an ozone layer, but far below modern levels. During this interval, Earth experienced the first of the major Snowball Earth glaciations of the Neoproterozoic, although the best dated and most extreme ones occurred later, ~720–635 Ma. The Huronian glaciations, ~2, for 2.1 Ga, straddle the GOE and may have been triggered by methane drawdown and are enhanced silicate weathering following continental assembly. Later, between ~1.0 and Ga, several poorly dated glacial intervals are recognized on some continents. Tectonically, the Middle Proterozoic saw the assembly and breakup of the supercontinent Rodinia, ~1, 30.9 Ga. Rodinia's configuration is still debated, but most reconstructions place Laurentia, ancestral North America, at or near the center, surrounded by Australia, Antarctica, India, Siberia, and the Proto-African and South American blocks. The assembly of Rodinia involved massive mountain building, Grenville orogeny in Laurentia, Sveka Norwegian in Baltica, etc., which may have enhanced silicate weathering, drawing down CO2 and contributing to later cooling. The boring billion earned its name because eukaryotic diversification was slow, no animals or complex ecosystems appeared, and major geochemical and climatic indicators changed only gradually. Yet this apparent stability was deceptive. The eukaryotic cell had evolved, mitochondria and plastids had been acquired, sexual reproduction had appeared, simple multicellular algae existed. All of the genetic and developmental toolkit necessary for complex multicellular life was gradually being assembled, even though the environmental triggers and ecological opportunities for rapid radiation were not yet in place. That dramatic ecological and evolutionary shift, the second major oxygenation event, the extreme cryogenian glaciations, the appearance of the first macroscopic multicellular organisms, and finally the prelude to the Cambrian Explosion, we now enter the most dynamic and climatically extreme phase of the Proterozoic Eon, the Neoproterozoic era, spanning approximately 1,000 to 541 million years ago. This final chapter of the Precambrian is anything but boring. It is marked by extraordinary environmental upheavals, repeated near-global glaciations, the second major rise in atmospheric oxygen, the appearance of the first macroscopic multicellular organisms, the emergence of complex ecological interactions, and right at the very end, the unmistakable biological prelude to the Cambrian Explosion. This is the transitional world where the long microbial-dominated era finally begins to give way to something recognizably more complex. By tilde 800 million years ago, several key preconditions for complex life were already in place. Eukaryotic cells with mitochondria and in the photosynthetic lineages, plastid sexual reproduction with meiosis and syngamy, allowing greater genetic diversity and more rapid evolutionary adaptation, simple multicellularity in several algal groups, red algae, green algae, possibly brown algae, low but non-zero atmospheric oxygen, tilde 110% PAL, a protective ozone layer, and surface waters that were at least intermittently oxygenated, yet the oceans remained predominantly anoxic below the mixed layer, with widespread euxinia, free hydrogen sulfide, in many intermediate and deep water settings. This limited the vertical extent of oxygenated habitats and kept ecological complexity low. Then, beginning around 720 million years ago, Earth entered one of the most dramatic climatic episodes in its entire history, the cryogenian glaciations, often referred to as Snowball Earth events. Two major intervals are recognized: Sturtian glaciation, tilde 720-660 Ma, Marinone glaciation, tilde 650-635 Ma. During these episodes, especially the Marinone, geological evidence from every continent, including low-latitude equatorial deposits, shows that ice extended to or very near sea level even at the equator. Dropstones, glacial tillites, cap carbonates, and iron formations deposited beneath ice are found in Australia, Namibia, Canada, China, Brazil, and elsewhere. The Snowball Earth hypothesis proposes that runaway ice albedo feedback drove the planet into a fully or nearly fully glaciated state. Once ice reached 40 degrees latitude, increased reflection of sunlight caused further cooling, advancing ice toward the equator. Open water areas, if any, were restricted to narrow equatorial belts or persistent leads in sea ice. Average global temperatures may have dropped to 50 degrees C or lower at the surface, with thick sea ice, perhaps 1 km or more, covering the oceans. Photosynthesis would have been severely restricted. Most marine primary production likely retreated to thin bands of open water or to cracks and melt ponds on the ice surface. Many microbial lineages probably went extinct or survived only in refugia, hydrothermal vents, subglacial lakes, brine channels, and sea ice. How did Earth escape this frozen state? The leading mechanism involves the long-term carbon cycle. Even under ice-covered conditions, volcanism continued to release CO2 into the atmosphere. With no liquid water to weather silicate rocks and draw down CO2 through the formation of carbonates, volcanic CO2 accumulated over millions of years until the greenhouse effect became strong enough to melt the ice. Once melting began, ice albedo feedback reversed catastrophically. Dark ocean water absorbed sunlight, accelerating warming. Massive carbonate precipitation formed the characteristic cap carbonates that blanket glacial deposits worldwide, a geochemical signature of rapid CO2 drawdown after deglaciation. These extreme glaciations may have been triggered by the breakup of the supercontinent Rodinia, tilde 750-700 Ma, which increased continental margins, enhanced silicate weathering initially, lowering CO2, and altered ocean circulation. Other factors, possibly a faint young sun, changes in methane production, or large igneous provinces, may have contributed. The biological consequences of the cryogenian glaciations were profound. The post-Marinone world, tilde 635 Ma onward, shows a rapid rebound in eukaryotic diversity. Within just a few million years after the last ice melted, we find the first macroscopic multicellular organisms. The earliest and most famous of these are the Ediacara biota, or Ediacaran organisms, named after the Ediacara Hills in South Australia where they were first described in the 1940s. They appear in the fossil record shortly after ~635 Ma and persist until ~541-538 Ma, right up to, and in some places slightly across, the Ediacaran-Cambrian boundary. Ediacaran fossils are preserved in exceptional soft-bodied conservat Lagerstätten, e.g., Ediacara Hills, Mistaken Point in Newfoundland, White Sea in Russia, Nama Group in Namibia. They include disc-shaped and frond-like forms, Dickinsonia, Spraguina, Charnia, quilted mattress-like bodies with serial or fractal-like modules, tube-like and bag-like structures, some forms with possible holdfasts, stems and fronds resembling modern sea pens. Their biology remains controversial. Interpretations include early animals, Cnidarian grade, early bilaterians, vendobionts, a separate kingdom of quilted, multinucleate organisms unrelated to modern phyla lichen like symbioses between algae and fungi, microbial colonies, or algal mats elevated above the sediment. Recent biomarker and isotopic evidence increasingly supports an animal affinity for at least some ediacarans, e.g., cholesterol biomarkers in Dickinsonia fossils. Possible muscle-like tissues in some specimens. Regardless of exact affinities, these were the first large, morphologically complex multicellular organisms visible to the naked eye, some reaching 1–2 meters in length. At roughly the same time, ~600-550 Ma, small shelly fossils, SSFs, begin to appear, tiny mineralized tubes, spines, scales, and sclerites made of calcium phosphate or calcium carbonate. These represent the earliest evidence of biomineralization on a widespread scale: shells, spicules, and protective structures. Many are likely skeletal elements of small animals, e.g., early mollusks, brachiopods, sponges, annelids, though most cannot be confidently assigned to living phyla. Trace fossils also become more abundant and diverse in the late Ediacaran. Simple burrows, trails, and grazing traces appear, indicating that motile organisms were actively disturbing sediments, something microbial mats had largely prevented for billions of years. Atmospheric oxygen rose again during the late Neoproterozoic, possibly reaching 10–20% PAL or higher by ~550 Ma. This Neoproterozoic oxygenation event is recorded by increased redox-sensitive trace metals, Mo, Vu, in black shales, sulfur isotope shifts indicating higher seawater sulfate decline in banded iron formations, more widespread oxic deep water conditions, higher oxygen likely permitted larger body sizes, more active metabolism, and the evolution of tissues that require aerobic conditions, e.g., collagen, muscle. By the very end of the Ediacaran, ~541 Ma, the stage was fully set. The oceans were richer in oxygen, biomineralization was underway, ecological relationships were becoming more complex, genetic toolkits for development and cell differentiation had evolved, and the first diverse communities of macroscopic organisms had appeared. The Cambrian Explosion, the rapid diversification of animal phyla with hard parts and complex ecologies, was imminent. We now stand at the very threshold where the long, patient silence of the Precambrian finally gives way to the unmistakable clamor of animal life. The boundary between the Ediacaran period, the final chapter of the Proterozoic, and the Cambrian period, the opening act of the Phanerozoic Eon, is dated to 541 million years ago. This thin line in the rock record, defined globally by the first appearance of the trace fossil Treptechnus pedum, marks one of the most extraordinary transitions in the entire history of life on Earth, the Cambrian Explosion. For more than 3 billion years, life had been almost exclusively microbial. Even in the latest Ediacaran, macroscopic organisms were strange, soft-bodied, mostly sessile or drifting forms whose exact relationships to modern animals remain debated. Then within a geological instant, perhaps 10 to 25 million years, possibly even less, nearly all the major animal body plans, phyla, that exist today appeared for the first time in the fossil record. Shells, skeletons, articulated limbs, eyes, mouths, digestive tracts, nervous systems, and complex ecological interactions burst onto the scene with astonishing rapidity. This sudden diversification is what we call the Cambrian Explosion or Cambrian radiation. It is not that animals themselves originated at that moment. Molecular clock estimates and a handful of Ediacaran biomarkers suggest that the earliest animals, probably sponge grade or cnidarian grade, already existed in the Cryogenian or early Ediacaran. Rather, the explosion refers to the geologically abrupt appearance of readily fossilizable, morphologically complex, ecologically diverse animals with hard parts and active lifestyles. Key features of the Cambrian explosion include rapid appearance of phyla by the end of the early Cambrian, 514–509 Ma, stage 3–4. Most major bilaterian phyla are represented: arthropods, mollusks, annelids, brachiopods, echinoderms, chordates, early vertebrates, priapulids, lobopodians, and many others. Even phyla that today have no mineralized hard parts, e.g., nematodes, flatworms, left trace fossils or exceptionally preserved soft-bodied remains. Biomineralization revolution: the small shelly fossils, SSFs, that began in the latest Ediacaran exploded in diversity and abundance across the early Cambrian. Animals began producing calcite, aragonite, calcium phosphate, and organic sclerites at an unprecedented scale. This was not a single evolutionary event, but a polyphyletic trend across many lineages, likely triggered by rising calcium and phosphate availability in seawater, ecological pressures, predation, and genetic toolkits already in place. Development of complex ecologies: for the first time we see clear evidence of tiered food webs, predators drilling holes in shells, animals burrowing deeply into sediment. Bioturbation reaches new depths and intensities: grazing, filter feeding, scavenging, and active swimming. Trace fossils diversify dramatically. Simple Ediacaran trails give way to branching burrows, resting traces, crawling tracks, and predation marks. Exceptional preservation windows: several famous Lagerstätten provide windows into the soft-bodied diversity that hard part fossils miss. These deposits preserve eyes with lenses, e.g., Anomalocaris, trilobites, gills, digestive tracts, nervous tissue, muscle fibers, and delicate appendages, revealing that early Cambrian animals were already anatomically sophisticated. Sirius Passet, Greenland, 518 Ma, Chengjiang, South China, 518 Ma, Emu Bay Shale, Australia, 514 Ma, Burgess Shale, Canada, 508 Ma. Why did the Cambrian explosion happen when it did? No single cause explains it fully. Most researchers now favor a confluence of multiple environmental, ecological, and genetic factors. Second major oxygenation event: atmospheric O2 likely rose to 10–20% PAL or higher by 550–520 Ma. Deep ocean anoxia declined in many regions. Higher oxygen supported larger body sizes, higher metabolic rates, collagen-based tissues, and active predation. Ecological feedback loops: the appearance of predators created intense selection pressure for protective shells, spines, and escape behaviors. This arms race drove rapid morphological evolution. Burrowing and bioturbation oxygenated sediments, creating new niches and recycling nutrients. Nutrient and chemical changes increased continental weathering, possibly linked to Rodinia breakup and assembly of Gondwana, delivered more phosphorus, calcium, and trace metals to the oceans, enabling biomineralization. Genetic and developmental toolkit: Hox genes, signaling pathways, WNT, hedgehog, TGF-beta, and other developmental control genes had already evolved in simple form in early metazoans. Small genetic changes could now produce large morphological effects, facilitating evolutionary experimentation. Extinction of Ediacaran incumbents: many Ediacaran taxa disappear at or shortly before the Ediacaran-Cambrian boundary. Whether due to environmental stress, competition, predation, or ecological displacement, their decline may have opened ecospace for the new bilaterian fauna. By the Middle Cambrian ~509-500 Ma, the seas teemed with trilobites of every size and shape, brachiopods filtering water, hyoliths and mollusks crawling or swimming, priapulid worms burrowing, anomalocaridids hunting with grasping appendages and compound eyes, early chordates, Pichia, Yunnanozoon filtering food in the water column, and sponges, archaeocyaths and calcimicrobes building the first animal-constructed reefs. The land remained silent. No plants, no arthropods, no vertebrates had yet ventured ashore. Microbial mats still covered exposed rock in places, but true soils and terrestrial ecosystems were still hundreds of millions of years in the future. The Cambrian Explosion was not the origin of animals, but the moment when animals became ecologically dominant, morphologically diverse, and readily preservable in the rock record. It marks the true end of the before animals world. From this point forward, Earth would never again be a planet ruled solely by microbes. The quiet microbial Earth had given birth to a new dynasty, one that would eventually produce forests, coral reefs, dinosaurs, mammals, and eventually ourselves. We have now traveled through the immense sweep of time from the molten birth of Earth to the very edge of the animal world. Before we cross fully into the Phanerozoic and watch the Cambrian seas explode with trilobites, brachiopods, anomalocaridids, and the first clear predators with compound eyes and grasping appendages, it is worth pausing, really pausing, to absorb the scale and character of everything that came before. Because the single most striking truth about Earth before animals is not any particular event, not the Great Oxidation Event, not Snowball Earth, not even the quiet arrival of the first eukaryotic cells. The single most striking truth is duration. Animals, in the sense of motile, heterotrophic, multicellular organisms with tissue-level organization and nervous systems, have existed for perhaps 600–750 million years at most. That sounds like an extremely long time to a human mind. Yet when placed against the total age of Earth, it is startlingly brief. Earth's age, 4–540 million years, first convincing animal traces, approximately 560–580 Ma, first unambiguous bilaterian animals with hard parts. Approximately 541–520 Ma, therefore, animals occupy approximately 11–13% of Earth history. More precisely, complex macroscopic animals with mineralized skeletons and predator-prey dynamics occupy approximately 11.5% of Earth history. That means for roughly 88–89% of the time that this planet has existed as a recognizable geological body, there were no animals. No footsteps on wet sediment, no burrows churning the seafloor, no shells littering beaches, no jaws crushing exoskeletons, no schools of fish-like forms flashing silver in the light, no reefs built by the metabolic activity of heterotrophs, no sound of anything that could be called feeding, fleeing, mating, hunting, grazing, drilling, escaping. Only chemistry, physics, geology, hydrology, and for the final third of that immense interval, microbiology. That is the central emotional and intellectual fact we must hold in our minds if we want to feel what before animals really means. It is not merely an absence of certain body plans. It is an absence of an entire mode of being. The planet ran on a completely different economy of energy and information for billions of years. Let us try, therefore, to hold several simultaneous images in our awareness at once, images that are difficult to keep together, because they are so qualitatively different from the animal-saturated world we inhabit. Image 1: the sheer patience of the Hadean and early Archean. For the first 600–800 million years after Earth formed, the surface was either molten or repeatedly sterilized by impacts energetic enough to vaporize oceans. When liquid water finally became stable and persistent, perhaps around 4.4–4.3 billion years ago, there was still no life, or if there was, it left no unambiguous trace that has survived. The planet had to wait through hundreds of millions of years of purely geochemical cycling before even the simplest self-replicating molecular systems could emerge and persist. That waiting period is longer than the entire existence of complex animals up to the present day. Image 2: the microbial world in full maturity between roughly 3.5 billion and 800 million years ago, the dominant form of life. Really, the only abundant, ecologically important form of life was microbial. The oceans, tidal flats, hydrothermal systems, anoxic basins, and very late in that interval, perhaps moist soil surfaces were covered in layered microbial mats. Stromatolites were the most conspicuous macroscopic structures produced by biology for billions of years. They were not built by animals, they were built by cyanobacteria, other photosynthetic bacteria, sulfate reducers, iron reducers, methanogens, and many other metabolic guilds living in close spatial and chemical cooperation. These mats were the forests, grasslands, coral reefs, and kelp beds of the Precambrian world. They were the primary producers, the sediment stabilizers, the oxygen factories, after Ga, the carbon sinks, the nitrogen fixers. The biomass and productivity of the late Archean and Proterozoic Earth were overwhelmingly microbial. Image 3: a planet running on redox disequilibrium without predation for almost the entire history of life before animals. Ecosystems were not structured around predator-prey relationships. Energy flowed mostly through one-step or very short food chains. Primary producers, cyanobacteria, anoxygenic phototrophs, chemolithotrophs, right arrow grazers, or direct consumers of dissolved organics or dead cells, right arrow decomposers returning nutrients. There was no trophic level devoted to animals that actively hunt, capture, kill, dismember, and digest other large multicellular organisms. The metabolic rate of the biosphere was therefore far lower per unit biomass than it became after the Cambrian. Most energy was dissipated very close to where it was captured. Image 4: the silence. This point is almost impossible to overstate. The Precambrian Earth was radically silent in biological terms. No stridulation, no birdsong, no whale song, no frog chorus, no insect buzz, no footfalls, no chewing, no crunching shells, no scraping gradually, no rasping of teeth on bone. The only sounds were abiotic: waves breaking on empty basalt shores, wind moving across bare rock or microbial mats, distant thunder from volcanic plumes or large storms, the low-frequency groan of tectonic plates grinding against one another, the occasional crack of cooling lava or ice. For billions of years, the only music of life was the inaudible chemical conversation between cells, proton gradients, electron flows, diffusible signaling molecules, and the gentle fizz of oxygen bubbles escaping cyanobacterial mats on a sunny afternoon in shallow water 2.7 billion years ago. Image 5: the slow construction of preconditions. Every major precondition required for animal life took hundreds of millions to billions of years to assemble. Reliable liquid water, approximately 4.4 Ga self-replicating systems, approximately 3.8 Ga oxygenic photosynthesis, approximately 2.7 Ga persistent atmospheric oxygen and ozone, approximately 2.4 Ga onward eukaryotic cell plan. Approximately 1.85–1.6 Ga plastid endosymbiosis in the green and red lineages. Approximately 1.6–1.2 Ga sexual reproduction, before 1.2 Ga simple multicellularity in several lineages. Approximately 1.20–0.8 Ga second major oxygenation pulse. Approximately 850–540 Ma collapse of widespread euxinia in shelf seas. Approximately 580–540 Ma ecological vacancy created by Ediacaran extinction filter. Approximately 550–541 Ma each step depended on the previous ones. Remove any one of them and the trajectory toward animals either halts or is delayed by geological timescales. The Ediacaran Cambrian transition, therefore, was not the moment animals were invented. It was the moment when all the slow-built preconditions finally aligned with an ecological opportunity large enough, and an oxygen level high enough, and a developmental toolkit flexible enough, that evolutionary experimentation could produce dozens of distinctly different body plans in a geological blink. That is why the Cambrian explosion looks so sudden in the rock record. It is not that evolution suddenly became fast. It is that the ecological release was enormous and the genetic developmental machinery was finally ready to respond at speed. And yet even after the explosion, the animal-dominated world we think of as normal is still only the last 12% or so of the story. The other 88% was a different planet, not merely a planet waiting for animals, but a planet living a full, complete, self-sufficient existence without them. Let us slow down even further now. We have already passed through the fire of planetary accretion, the long wait for the first stable oceans, the patient microbial takeover that lasted longer than most mountain ranges endure, the quiet oxygenation of a world that did not ask to breathe oxygen, the repeated near-death experiences of global ice cover, the strange soft-bodied experiment called the Ediacara biota. And finally, the geological instant when mineralized hard parts and active predation became ecologically unavoidable. And yet, after recounting all of that, the single sentence that still feels most honest is this one. For more than 4 billion years, Earth did not need animals to be a living planet. It functioned beautifully, even extravagantly, without them. That sentence deserves to be unpacked slowly, layer by layer, because it runs so deeply counter to the instinctive animal-centric storytelling most of us inherit. We tend to imagine deep time as a long preparation, a kind of cosmic preamble whose only real purpose was to reach the moment when legs, eyes, jaws, brains, and societies finally appeared. But that is retrospective storytelling. From the perspective of any given moment between, say, 3.5 billion and 800 million years ago, the world already looked complete. It had producers, consumers, decomposers, geochemical cycles that balanced themselves over eons, stable enough redox gradients to support persistent disequilibrium, the very thing life exploits, self-regulating feedbacks involving biology and geology. It had beauty of its own kind. The slow rise of stromatolitic reefs along ancient coasts under an orange-tinted sky, the subtle color shifts in banded iron formations as oxygen first learned how to stain a planet red, the patient formation of black shales in anoxic basins where organic carbon was entombed for hundreds of millions of years, the silent precipitation of manganese nodules on abyssal plains, the rhythmic layering of varved sediments in proglacial lakes during the Huronian ice ages. None of those phenomena required an animal to witness them, to eat them, to build a shell from them, to swim through them, to evolve camouflage against them. They simply were. So let us try, however imperfectly, to inhabit the point of view of that long animal-free interval. Imagine you are a cyanobacterium living in a shallow lagoon 2.7 billion years ago. The water above you is warm, perhaps 40-50 degrees C, supersaturated with respect to calcium carbonate. Sunlight filters through a hazy CO2-rich atmosphere. The wavelengths that reach you are slightly shifted toward the red end of the spectrum because Rayleigh scattering is weaker without much oxygen or ozone. You are part of a layered mat a few millimeters thick. At the very top live your closest relatives, other filamentous and unicellular cyanobacteria using photosystem II to split water and release oxygen. A few micrometers below you, the oxygen concentration drops to zero within seconds. Anaerobic sulfur bacteria and methanogens take over, using the organic matter you and your neighbors exude during the day. Below them again are sulfate reducers producing hydrogen sulfide that diffuses upward and poisons any oxygen that tries to penetrate too deeply. Your entire ecosystem is compressed into less than a centimeter of vertical space, yet it contains multiple redox zones, multiple guilds, multiple ways of harvesting energy. You do not have a brain, eyes, muscles, or even flagella, although some of your cousins do. You do not hunt, you do not flee. You photosynthesize when light is available and respire or ferment when it is not. When you die, your cell wall contributes a tiny increment to the organic matrix that traps the next layer of sediment. Over centuries, your mat, and thousands like it, builds a demal stromatolite that rises half a meter above the surrounding seafloor. In a million years, that structure may be part of a kilometer-thick carbonate platform visible from space, if anything with eyes had existed to look. From your perspective, the world is already rich, complex, dynamic, complete. There is no sense of waiting for anything more advanced. The future does not exist as a felt absence. Now shift perspective again, this time to the deep ocean floor at billion years ago, shortly after the first convincing eukaryotic cells appear in the fossil record. You are now an aggregate of iron-manganese oxyhydroxides forming part of a slowly growing polymetallic nodule. Around you, the water is cold, dark, almost still. The only light is the faint chemiluminescence of some distant vent fauna. If such things already existed, we are not sure. Dissolved oxygen is vanishingly low. Nitrate, manganese, Roman 4, iron, Roman 3, and sulfate serve as the dominant electron acceptors. The primary energy source for the sparse microbial community living in and on the sediment is the very slow oxidation of organic carbon that rained down from the distant sunlit surface. Perhaps 1 or 2% of surface primary production actually reaches this depth. Most of what arrives is already highly refractory. Here, too, the system feels complete. There are iron oxidizers, manganese oxidizers, organoheterotrophs, sulfate reducers, methanogens living in consortia so tight that one cell's waste product is the next cell's substrate. The nodule grows by about 1 atomic layer per century. In 10 million years it might reach the size of a grapefruit. Nothing rushes, nothing hunts. The redox tower is long and stable. One more shift, this time to the height of the Marinone Snowball at 640 million years ago. The ocean beneath you is sealed beneath several hundred meters of sea ice. Photosynthesis is restricted to cracks, melt ponds, thin ice-covered leads, perhaps to pockets of liquid brine within the ice itself. Primary production is probably 1 or 2 orders of magnitude lower than during non-glacial intervals. Most of the biosphere has retreated to hydrothermal vents on the seafloor where chemistry, not sunlight, provides energy. Hydrogen, methane, sulfide, ferrous iron, manganese flow from black and white smokers. Methanogens, acetogens, sulfur reducers, iron reducers, nitrate reducers, perhaps already some aerobic methanotrophs and sulfur oxidizers form dense biofilms around the vent orifices. Temperature gradients of 300 degrees C over a few centimeters support dozens of metabolic guilds within a few square meters. Again, even in this near-apocalyptic state, the world is not empty. It is populated, metabolically diverse, chemically dynamic. The planet has not stopped being alive, it has merely changed address. Life has moved to the one place where thermodynamics still allows it to persist, the interface between hot rock and cold seawater. These 3 vignettes, the sunlit microbial mat, the dark abyssal nodule community, the sub-ice vent refuge, are not exceptions, they are typical. For the overwhelming majority of Earth history, wherever there was a sustained energy gradient and liquid water, or at least brine, life was present, doing what life does, extracting free energy, maintaining far from equilibrium states, reproducing with variation, slowly changing over geological time. The arrival of animals did not complete the planet. It changed the rules of the game. It introduced a new way of dissipating energy gradients through active motility, predation, herbivory, bioturbation, ecosystem engineering, but it did not replace the older ways. Even today in 2026, the biomass of prokaryotes in subsurface sediments, oceanic crust, and continental groundwater is estimated to rival or exceed the biomass of all animals combined. The oldest and most fundamental economy of the biosphere is still microbial. So when we say before animals, we are not describing an impoverished prelude. We are describing a different kind of richness, one measured not in phyla or body sizes or food chain lengths, but in metabolic versatility, geochemical persistence, temporal endurance. That realization carries a strange emotional aftertaste. On one hand, it is humbling. Our entire lineage, every vertebrate, every arthropod, every mollusk, every annelid worm, is a very late, very narrow experiment within a system that was already mature and self-sustaining for billions of years before the first sponge-like creature learned to filter feed. On the other hand, it is oddly comforting. The planet did not need us or anything like us to become beautiful, complex, chemically inventive, geologically active, climatically regulated. It managed all of that with nothing more than cells a few micrometers across, operating on the redox potentials available in water, rock, and sunlight. That long microbial prelude also changes how we read the Cambrian Explosion. It stops being the moment life finally got interesting and becomes something closer to the moment one particular metabolic guild, phagotrophic eukaryotes, discovered a new and explosively effective way of making a living. Animals are not the culmination of Earth history. They are a highly successful evolutionary radiation that happened to occur after all the hard preconditions had already been met. In the remaining parts of this narration, we will begin to draw the threads together, looking at what the animal-free Earth can teach us about resilience, about alternative biologies, about the deep strangeness and familiarity of our own planet, before finally returning to the quiet, almost tender moment when the first unambiguous animal traces appear in the rock and the long silence ends. But for now, let the images linger. A billion-year-old stromatolite dome catching the first reddening light of a hazy dawn, a fist-sized manganese nodule resting in darkness a kilometer below the surface, growing one atomic layer per century, a black smoker chimney under ice, surrounded by microbial veils undulating in warm vent fluid while the world above is locked in eternal winter. All of them alive, all of them sufficient. We have spent many hours now walking together through the long corridors of time before animals appeared. We have seen lava oceans glowing beneath a debris-choked sky, zircon grains locking away the memory of the first raindrops 4.5 billion years ago, microbial mats building domes in orange-tinted shallows, while the rest of the planet remained geologically adolescent, banded iron formations growing layer by layer like tree rings of a world learning how to breathe oxygen, snowball episodes in which ice reached the equator and life clung to the warm throat of volcanic vents. Strange quilted ediacaran bodies lying on microbial felt like leaves fallen on damp forest floor, and then right at the boundary, the first unmistakable scratch marks of small worm-like bodies moving purposefully through wet sediment. Through all of it, one sensation keeps returning, stronger each time: scale. Not just size, although the numbers are already incomprehensible, but temporal scale, the sheer moral weight of duration. Let us try, therefore, to make that scale feel less abstract and more bodily. Take the entire duration from the Cambrian Explosion until today, roughly 541 million years, and imagine it compressed into a single 24-hour day. Midnight right arrow beginning of Cambrian approximately 4:00 a.m. right arrow first fish like vertebrates appear approximately 10:00 a.m. right arrow first forests on land. First insects approximately 2:00 p.m. right arrow first dinosaurs approximately 10:50 p.m. right arrow non-avian dinosaurs go extinct approximately 11:40 p.m. right arrow earliest anatomically modern humans appear approximately 11:50 p.m. right arrow beginning of agriculture approximately 11:59 8 p.m. right arrow start of the Industrial Revolution approximately now right arrow 00:00:00:2 of the next second. That entire rich, violent, beautiful animal story, from the first trilobite eyes to space telescopes and symphonies, occupies the final 0.2 of a second before midnight. Now rewind the clock and stretch the same 24-hour metaphor backward across the whole of Earth history. Midnight right arrow present day working backward, approximately 11:59 8 p.m. right arrow Industrial Revolution. Tiny sliver containing all recorded human history, approximately 11:40 40 p.m. right arrow first Homo sapiens approximately 10:50 p.m. right arrow end Cretaceous extinction approximately 2:00 p.m. right arrow first dinosaurs approximately 10:00 a.m. right arrow first land forests approximately 4:00 a.m. right arrow Cambrian explosion. Now keep going backward. The hand moves very slowly now. Approximately 3:00 a.m. right arrow first macroscopic Ediacaran fossils approximately 1:30 a.m. right arrow Marinone snowball deglaciation approximately 12:45 a.m. right arrow Sturtian snowball begins approximately 11:30 p.m. previous day right arrow first red algae. First simple multicellularity approximately 10:00 p.m. right arrow first eukaryotic cells with plastids approximately 8:00 p.m. right arrow first eukaryotic cells with mitochondria approximately 6:00 p.m. right arrow Great Oxidation Event completes approximately 4:00 p.m. right arrow first oxygenic photosynthesis approximately 2:00 p.m. right arrow first stromatolites. First convincing microfossils approximately 12:30 p.m. right arrow end of late heavy bombardment approximately 11:00 a.m. right arrow first oceans stable approximately 9:00 a.m. right arrow moon forming impact approximately 6:00 a.m. right arrow earth fully accreted. Magma ocean stage approximately midnight start of the 24-hour day. Right arrow solar system begins to condense from molecular cloud. Everything we call animal history occupies roughly the last 35-40 minutes of that long day. Everything before it, the Hadean, Archean, most of the Proterozoic, occupies the first 23 hours and 20 minutes. That means a human being who lives to 85 has personally experienced about 0.003% of the planet's lifetime. A redwood tree that reaches 30 years has lived through 0.07% of Earth time. Even the oldest known continuously living clonal organism, a Tasmanian King's Lamatia shrub nicknamed Old T. Jicko, whose root system is approximately 9,500 years old, has existed for roughly 0.02% of Earth's age. Microbial lineages that have persisted since the Archean have experienced essentially the entire day. When we speak about Earth before animals, we are therefore speaking about the planet during the hours when almost everything important about its geochemical personality, its climate self-regulation, its biological toolkit, its redox architecture was already established, and all of it happened without nervous systems, without motility at macroscopic scale, without predation pressure driving arms races, without eyes looking back at the sun. That realization can produce several different responses at once. One is existential vertigo, the sudden understanding that our entire experiential world of movement, color perception, hunger, fear, courtship, parenthood, storytelling, music, war, love, grief, hope, science, art, religion, technology is not only late but minuscule in temporal proportion. Another is a kind of relief. The planet did not spend 4 billion years waiting to become interesting. It was interesting the whole time, just not in a way that produces headlines or museum displays. It was interesting in the way a slow coral atoll is interesting, or a mature boreal forest is interesting, or the slow carbon cycle through basalt weathering is interesting, complex, self-regulating, beautiful at scales humans rarely notice. A third feeling is gratitude of a peculiar kind. Because the long microbial prelude gave us oxygen levels high enough to support large active bodies, phosphate concentrations high enough to build bones and shells, ecological vacancy created by repeated extinction filters, and most mysteriously of all, a developmental genetic toolkit, Hox clusters, signaling pathways, cell adhesion molecules, apoptosis machinery, that was already largely in place before the first obvious animal appeared. The animal body plan did not have to be invented from scratch in the Cambrian. It was assembled from parts that had been slowly refined over eons in lineages that never became animals themselves. So the animals did not win the planet. They inherited a world that had already been tuned, oxygenated, fertilized, ecologically stress-tested, geochemically buffered. They were very late tenants moving into a house that had stood empty and well-maintained for an inconceivably long time. That metaphor, the house, is worth lingering on. Imagine inheriting a vast old estate that has stood for 4 billion years. The foundation was poured when the planet was still molten, Hadean accretion and differentiation. The plumbing was installed when hydrothermal vents first began circulating seawater through hot rock, origin of life chemistry. The electrical system was wired when cyanobacteria learned how to split water, oxygenic photosynthesis. The insulation and climate control were gradually improved as oxygen accumulated and an ozone layer formed, GOE, and subsequent redox evolution. The garden was planted and tended for billions of years by microbial communities that built soils, cycled nutrients, stabilized sediments, precipitated carbonate platforms kilometers thick. The library already contained almost the entire developmental genetic code needed to build complex multicellular forms. It was just that nobody had yet used those books to write very large-bodied stories. Then, in the last 12 minutes of the fourth day, a new group of tenants arrives, noisy, mobile, hungry, inventive, destructive, creative. They immediately begin redecorating, digging deep burrows that churn the old garden soil, building reefs that rival the ancient microbial platforms, cutting down parts of the forest to make room for pastures and cities, burning the fossil library pages to power machines. The original caretakers, the microbes, do not disappear. They continue to run the water treatment plant, the waste recycling center, the atmospheric regulation system, the nutrient loops in the soil, the base of every food chain. They remain the majority population by cell number and by total living biomass in most environments. The new tenants are spectacular, but they are still minority shareholders in the enterprise. Seen in that light, the phrase "before animals" stops sounding like an empty waiting room. It begins to sound more like the time when Earth managed its own affairs perfectly well, thank you very much, and laid down almost everything necessary for the brief, brilliant, noisy animal interlude that followed. That interlude is still ongoing, we are living inside it, but it is already clear that it will be brief compared with what came before. Whether it ends in 5 million years or 500 million years, the animal-dominated surface biosphere will almost certainly constitute less than 15% of the total lifetime of the planet. Afterward, whether the cause is runaway greenhouse, large impact sterilization, slow loss of oceans to photodissociation and hydrogen escape, or something else entirely, the planet will very likely return to a state much closer to the one it knew for most of its existence. Predominantly microbial, quiet at macroscopic scales, chemically inventive, geologically active, still cycling carbon, nitrogen, sulfur, phosphorus, still maintaining redox disequilibrium across the surface and subsurface. In other words, after animals, Earth will probably once again become, in broad metabolic and geochemical outline, something very like the Earth that existed before animals. That circularity is strangely satisfying. It suggests that animals were never the goal. They were a phase spectacular, emotionally intense for those of us inside it, ecologically transformative, but still only a phase. The planet has done without them for 88% of its history and will very likely do without them, or with only sparse microbial regrowth, for most of whatever time remains before the sun expands into a red giant and boils the oceans away for good. Knowing that changes the emotional tone of the whole story. It is no longer primarily a heroic march toward complexity and consciousness. It becomes something more modest, more patient, more ecological. A very long, very slow, very successful experiment in living within geochemical gradients. An experiment that has already succeeded many times over. An experiment that invented photosynthesis, nitrogen fixation, sulfur cycling, methane production, methanotrophy, animox, metal oxidation, metal reduction, biomineralization, eukaryotic cell architecture, sex, multicellularity, developmental patterning. all without ever needing legs, eyes, hearts, brains, or societies. Animals were simply one very flashy way of dissipating free energy. A successful way, yes. A beautiful way, yes. A tragic, hopeful, creative, destructive, storytelling way, yes. But not the only way, not even measured across 4.5 billion years, the dominant way. The dominant way has always been and most likely will always remain microbial, quiet, patient, persistent, almost invisible, almost immortal. That is the Earth we have been trying to feel again in these pages, not as a prelude, not as a deficiency, but as a complete and sovereign world that existed for billions of years on its own terms. In the final part, we will return one last time to the very last moments before the Cambrian, those thin Ediacaran beds where the first clear animal traces appear, and then let the narration come to rest, leaving the reader with the same sensation we hope to carry ourselves. A deep, quiet awe at a planet that spent the overwhelming majority of its existence being perfectly interesting without ever once needing to evolve anything that could run, bite, see, scream, love, remember, or dream. And so we arrive, almost reluctantly, at the very last pages of the story that can still truthfully be called Before Animals. The rock layers we are looking at now are thin, sometimes only a few meters thick, yet they contain one of the sharpest transitions the fossil record ever shows. Above them, trilobites marching in orderly files across bedding plains, brachiopods filtering the water column by the thousands, hyolith cones half buried in lime mud, small mollusks leaving sinuous trails, priapulid worms pushing through sediment like living drills, anomalocaridids with circular mouthparts and grasping frontal appendages hunting in the shallows. Below them, something quieter, softer, stranger, harder to classify. These are the beds of the latest Ediacaran, roughly 555 to 541 million years ago, and the organisms preserved in them do not yet look like the Cambrian world that follows so quickly after. We find Dickinsonia, oval, quilted, mattress-like bodies up to 1.4 meters long, often lying flat on microbial mats, sometimes showing faint contraction marks as though the organism had tried to move by rippling its body. Sprigina, narrow, segmented forms with a crescent-shaped head region, trailing behind them a series of overlapping lobes like the footprints of a very soft caterpillar. Charnia, tall fronds anchored to the sediment by a holdfast, branching in a fractal-like pattern that reminds some researchers of modern sea pens, although almost certainly unrelated. Aspidella, simple circular impressions, possibly holdfasts or resting traces. Eandromeda, spiral, 8-armed forms that look almost like jellyfish bells pressed into the mud. Yorgia philizune andiva, more quilted sheets and fronds with raised ridges and grooves that suggest some kind of internal fluid pressure or tissue differentiation. None of these organisms appear to have possessed mineralized hard parts, except possibly tiny calcified tubes in a few cases. Almost none show clear bilateral symmetry with obvious anterior-posterior differentiation, mouth-anus axis, or appendages designed for active locomotion. Many seem to have lived attached to the microbial mat's surface or partially embedded within it, absorbing nutrients osmotically or via symbiotic microbes, rather than actively feeding. For decades, paleontologists argued over what these forms actually were. Early interpretations placed them as early representatives of modern phyla, Dickinsonia as a primitive annelid or flatworm, Sprigina as a segmented proto-arthropod, Charnia as a cnidarian-grade polyp. Later, during the 1980s and 1990s, Adolf Seelascher proposed the radical alternative that the Ediacara biota belonged to an entirely separate kingdom, the Vendobionta, built on a completely different body plan. A quilted, air-filled or fluid-filled new structure without true tissues, guts, or motility organs, more akin to a giant lichen or an inflated test than to any animal. More recent work, especially biomarker studies published between 2018 and 2024, has swung the pendulum back toward an animal affinity taxa. Dickinsonia fossils from South Australia preserve molecules interpreted as cholesterol derivatives, sterols almost exclusively produced by animals in modern ecosystems. Trace fossils associated with some Ediacaran forms show limited but unambiguous movement. The growth patterns of certain fronds suggest tissue-level organization rather than mere colonial aggregation. The current consensus, as of early 2026, is therefore a compromised position. Some Ediacaran taxa, especially the frondose and discoidal forms, may belong to stem groups of modern phyla, possibly total group Cnidaria, Ctenophora, or very early Eumetozoans. Others, the classic quilted Dickinsonia-type organisms, may represent an extinct grade of early metazoans that experimented with large body size before the Bolletterian developmental toolkit became dominant. Still others may be unrelated to animals, giant microbial colonies, multicellular algae, lichen-like symbioses, or even fungal-grade organisms. What matters more than the exact phylogeny is the ecological picture these fossils paint. The latest Ediacaran seafloor was still dominated by microbial mats, thick, cohesive, leathery layers that covered almost every surface in shallow to mid-depth waters. These mats suppressed erosion, trapped fine sediment, and created a stable, low-energy environment where soft-bodied forms could grow to surprisingly large sizes without needing mineralized skeletons for support or protection. There is almost no evidence of predation. Drill holes are absent. Shell-crushing scars are absent. No grasping appendages or radula-like scraping marks appear. The few trace fossils present are simple, shallow, horizontal burrows and crawling trails, nothing like the complex three-dimensional galleries that appear immediately after the boundary. In other words, the latest Ediacaran world still feels like an extension of the long microbial Earth we have been describing throughout this narration. The new large multicellular forms are lying on top of the old microbial landscape rather than fundamentally restructuring it. They are passengers, impressive in size, novel in body plan, but not yet ecologically transformative. Then, within a few million years, sometimes within a single bed, everything changes. Treptachnus pedum appears, a zigzag burrow system with short side branches, indicating an animal capable of systematic probing and backtracking, the first trace fossil complex enough to be used as the global marker of the Ediacaran Cambrian boundary. Small shelly fossils explode in diversity, tubes, spines, scales, cones, plates, all made of calcium phosphate or carbonate. Bioturbation intensity increases dramatically. Sediments become thoroughly churned rather than finely laminated. Predatory drill holes appear in tubular fossils. Trilobite-like arthropods leave unmistakable trackways. The microbial mat world begins to retreat as active grazers and burrowers break it up. The long silence is over. Not because animals suddenly evolved, they had already been evolving, quietly, for tens or hundreds of millions of years, but because the preconditions finally aligned so perfectly that the ecological release became explosive. Oxygen high enough to support high metabolic rates and collagen-rich tissues, seawater chemistry, favorable to rapid biomineralization, ecological vacancy, created by whatever caused the disappearance or sharp decline of most Ediacaran taxa, developmental genetic circuits capable of producing complex segmentation, appendages, nervous systems, sense organs, positive feedback loops between predation pressure, defensive adaptations, increased motility, deeper bioturbation. Nutrient recycling. The Cambrian world that emerges is not merely animals plus everything that came before. It is a qualitatively different biosphere, one in which energy flows much more rapidly through longer food chains, in which information processing nervous systems becomes a major evolutionary currency, in which macroscopic movement reshapes landscapes at rates never seen before. And yet, even after that transition, the microbial engine never stops. Even in the modern ocean, prokaryotic cells outnumber animal cells by at least 10 to 1. Subsurface microbial biomass may exceed surface animal biomass. Every breath you take contains oxygen originally split by cyanobacteria billions of years ago. Every atom of carbon in your body has passed through microbial cycles uncountable times. The animal experiment, spectacular though it is, remains embedded within and dependent upon the ancient microbial economy that preceded it and will almost certainly outlast it. So when we close this narration, we do not close it with the first trilobite eye or the first grasping claw. We close it with the image that has accompanied us the entire way. A shallow Precambrian sea at sunset, a stromatolite dome rising just above the waterline, cyanobacterial filaments catching the last red light, releasing tiny bubbles of oxygen that rise slowly to the surface, while beneath the mat, sulfate-reducing bacteria exhale hydrogen sulfide that will tomorrow feed iron oxidizers at the redox boundary a few millimeters deeper, while far offshore in darker water, a single soft, quilted Ediacaran body. Perhaps the very last generation of its kind lies quietly on the felted surface, absorbing dissolved organics, and somewhere in the genetic memory of its cells, instructions are already present for Hox genes, for apoptosis pathways, for cadherin-mediated cell adhesion, for muscle-like contractile proteins, instructions that will, in only a few million years, be read in a radically new way. That moment, the last quiet moment before the first unmistakable animal scratch appears in the sediment, is where the story we have been telling truly ends. After that comes noise, motion, predation, beauty, tragedy, intelligence, cities, telescopes pointed at other worlds, symphonies written in notation invented by brains that evolved from nerve nets that evolved from diffuse nerve nets that evolved from nothing more than chemical gradients across membranes in a microbial mat 2.7 billion years earlier. But before that, before all of it, there was simply Earth, living, breathing, cycling, waiting for nothing. Existing perfectly well for 4 billion years, with nothing that could run toward the light or away from danger or toward another of its kind with intent. Just chemistry becoming biology, becoming ecology, becoming geology, becoming time. And that, finally, is what Earth was really like before animals. We have now told the story forward and backward, in layers and in vignettes, through fire and ice, through chemical whispers and microbial empires, through the long red rusting of oceans and the quiet quilted gardens of the Ediacaran seafloor. And yet even after 10 parts, even after more than 11,000 words already laid down, the feeling persists that we have only brushed the surface, not because the facts are missing, but because the sensation of 4.5 billion years without animals is so qualitatively alien to every intuition we carry inside animal bodies. So let this 11th and final part be less a continuation of chronology and more a deliberate lingering, a refusal to hurry away from the silence we have been trying to hear. Let us sit, therefore, in one single imagined place for a very long time. Choose any shallow Precambrian tidal flat between 2 billion and 800 million years ago, say 1.4 billion years ago, midway through what is sometimes called the boring billion. The water is warm, perhaps 35-45 degrees C, slightly hypersaline in the upper intertidal zone where evaporation concentrates ions. The sky is a deep hazy amber. There is still no ozone shield thick enough to scatter blue light aggressively, so the sun appears larger and redder than today, its light arriving softened and diffused through a thick CO2-CH4 atmosphere. You are lying face down in a few centimeters of water, cheek pressed against a microbial mat that feels like wet velvet or very fine felt, slightly rubbery, slightly gritty from trapped carbonate grains and silicate silt. This mat is not uniform. In the topmost 0.10.3 live the dominant primary producers, unicellular and filamentous cyanobacteria whose sheaths are stained purple-brown by cytomin and other UV protective pigments. They are splitting water right now, under the midday light, releasing microscopic bubbles of oxygen that rise past your cheek and pop silently at the air-water interface. A few micrometers below that oxygenated skin, the oxygen concentration collapses to zero. Here live green sulfur bacteria and purple sulfur bacteria using bacteria chlorophyll to harvest longer wavelengths that penetrate deeper. They oxidize hydrogen sulfide, diffusing upward from even deeper layers. Below them again, perhaps half a millimeter down, sulfate-reducing bacteria respire anaerobically, producing more H2S that sustains the sulfur oxidizers above. Scattered throughout are methanogens quietly converting the last scraps of organic matter into methane that will eventually escape to the atmosphere and help keep the greenhouse effect strong enough to prevent runaway glaciation under the faint young sun. If you could shrink yourself to bacterial size and walk vertically downward through this mat, you would cross half a dozen redox zones in less than a centimeter, each zone a different metabolic kingdom, each one feeding off the waste of the one above, each one maintaining the disequilibrium that keeps the whole system running. The mat is not a community in the loose human sense of the word. It is a tightly coupled chemical factory in which every waste stream is someone else's feedstock. Now lift your head slightly. Around you the mat extends for tens to hundreds of meters in every direction, a continuous living carpet interrupted only by small channels where tidal currents have cut drainage rills, or by low stromatolitic domes that have grown upward over decades or centuries as sediment trapping and carbonate precipitation slowly elevate them above the surrounding flat. Some domes are smooth and hemispherical, others have cauliflower-like surfaces, still others have developed conical shapes with axial furrows where gas escape has focused upward flow. Touch one of these domes. The surface is slightly rough, like wet sandpaper, because each cyanobacterial filament is coated in extracellular polymeric substances, EPS, sticky mucilage that binds grains and prevents them from being washed away during the next high tide. Over hundreds of years, these incremental additions of sediment plus carbonate plus organic remains build structures that can reach half a meter or more in height. In some places, entire platforms kilometers across have formed this way, their internal lamination recording seasonal or tidal cycles in fine detail, dark organic-rich layers for wet seasons or neap tides, lighter carbonate-rich layers for dry seasons or spring tides. There is no sound except the soft lap of water against the matte surface, the occasional fizz of an oxygen bubble breaking, the distant low-frequency rumble of a far-off thunderstorm over the open ocean. No birds wheel overhead, no crabs scuttle sideways, no fish flash silver in the shallows, no insect wings catch the light. The only motion is the slow pulsing of water in and out with the tide, the subtle expansion and contraction of gas bubbles trapped beneath the mat, the imperceptible upward growth of the stromatolites themselves. If you stay in this place for a full year, you will witness almost no macroscopic change. The mat thickens by a fraction of a millimeter. A few new small domes appear where nucleation sites, perhaps a pebble or a ripple crest, allowed initial sediment trapping to begin. Methane seeps slowly from the anoxic subsurface and escapes to the atmosphere. Oxygen produced during the day mostly diffuses downward and is consumed at night by respiration within the mat itself. Only a tiny fraction escapes to the overlying water column. If you stay for 1,000 years, the largest stromatolites have grown noticeably taller. The platform has prograded a few meters seaward or landward, depending on sediment supply and relative sea level change. The isotopic composition of the carbonates has recorded subtle shifts in global carbon cycling. If you stay for 10 million years, entire formations have been deposited, hundreds of meters of laminated dolomite and limestone whose fine layering still preserves the memory of those ancient mats. The oxygen concentration in the atmosphere has ticked upward by a few parts per million. The redox state of the deep ocean has shifted slightly. Evolutionary tweaks have occurred within cyanobacterial genomes, perhaps allowing better UV protection or more efficient CO2 fixation. But at no point during those 10 million years does anything appear that would make you say, now the world has become interesting in the way I understand interesting. There are still no eyes looking back at the sun, no hearts pumping, no nerves firing, no behaviors that could be called courtship or territoriality or migration. The The system simply continues, self-regulating, self-repairing, self-perpetuating. Now let us do something emotionally difficult. Hold that image simultaneously with the knowledge that this same kind of microbial mat ecosystem dominated shallow marine environments for roughly 3 billion years, from Ga until the late Ediacaran, with only gradual modifications. 3 billion years of afternoons like the one we just imagined. 3 billion years of bubbles rising and popping in silence. 3 billion years of millimeter-scale redox towers turning sunlight and seawater into organic matter and oxygen and sulfide and methane. 3 billion years without a single organism ever once chasing another organism with intent to eat it. 3 billion years without anything ever once fleeing in terror. 3 billion years without anything ever once choosing a mate on the basis of visual display or song or dance or scent. 3 billion years without anything ever once building a nest, guarding young, teaching offspring, remembering a past, anticipating a future beyond the next tidal cycle. 3 billion years in which the most dramatic events from the perspective of anything living there were probably changes in salinity after a storm, or a pulse of hydrothermal fluid altering local pH, or a thin layer of volcanic ash temporarily shading the mat and causing a temporary dieback followed by recolonization. That is the texture of before animals. It is not barren, it is not boring, it is not waiting. It is a fully realized, fully successful, fully mature biosphere that happened to operate on spatial scales of micrometers to meters and temporal scales of hours to eons, rather than on the spatial scales of centimeters to kilometers and temporal scales of seconds to decades that feel normal to us. We find it hard to feel that maturity because our nervous systems evolved to notice exactly the kinds of events that were absent: sudden movements, sharp contrasts, intentional behavior, cause and effect chains that resolve in seconds or minutes. But the microbial Earth did not lack complexity. It simply expressed complexity in different currencies. In the number of metabolic pathways per cubic millimeter in the phylogenetic depth of lineages, coexisting in a single gram of sediment, in the stability of geochemical cycles maintained across hundreds of millions of years, in the sheer persistence of life through asteroid impacts, snowball episodes, supercontinent cycles, and massive volcanic outpourings. When we say the Cambrian Explosion was rapid, we mean rapid on a geological timescale. Tens of millions of years instead of hundreds of millions. But even that rapid interval is still longer than the entire Cenozoic era, the age of mammals, from the extinction of non-avian dinosaurs to today. The microbial world had already experienced hundreds of such rapid evolutionary pulses: the origin of oxygenic photosynthesis, the eukaryogenesis event, the plastid endosymbiosis, the invention of meiosis, each of which was probably just as transformative from the inside as the Cambrian radiation felt from the inside to early bilaterians. We simply don't see them as explosions because they didn't leave behind large skeletons or complex trace fossils. So the deepest lesson of Earth before animals may not be about absence at all. It may be about plenitude in forms we have been trained not to notice. It may be that the planet has spent most of its history demonstrating that life does not need to become large, fast, visible, motile, predatory, intelligent, or conscious in order to be abundant, resilient, diverse, inventive, beautiful, and enduring. All it needs is a sustained energy gradient, liquid water or brine, and time measured not in heartbeats but in half-lives of isotopes, in orbital cycles, in mantle convection rates. And given those three things, life will find a way, usually many ways, and then it will keep finding ways for billions of years without ever once needing to evolve anything that could look back at the stars and wonder where it came from. That finally is the quietest and perhaps the most startling truth we can carry away from this long journey. For the overwhelming majority of its existence, Earth was already a complete, successful, living world, and animals were never necessary. They were simply one more thing that happened, a beautiful, noisy, tragic, hopeful, creative, destructive, wondering thing, but still only one thing among many. And when the animal chapter eventually closes, whether in 5 million years or 500 million, the world will very likely fold back into something recognizably like the one that preceded us. Microbial mats quietly rebuilding in the shallows, stromatolites catching the next sunrise, bubbles of oxygen rising and popping in silence, redox towers turning over day after night after day after night, the long, patient chemistry continuing long after every last footprint has washed away. We cannot leave yet, even after 11 parts, even after the last quiet bubble of oxygen has risen and popped on a Precambrian afternoon, even after we have watched the first unambiguous animal trace fossil appear like a hesitant signature at the bottom of a contract the planet never asked to sign, there is still something unfinished. The unfinished thing is not more facts. We have recited enough dates, enough isotope ratios, enough names of formations and fossil genera. The unfinished thing is the feeling, the bodily, almost homesick feeling of what it actually meant to be alive on a planet where nothing ever ran, nothing ever hid, nothing ever chased, nothing ever chose, nothing ever mourned, nothing ever hoped. So this part will not advance the timeline. It will not introduce new events. It will simply stay much longer than feels comfortable, inside one single afternoon on one single tidal flat, somewhere between 2.5 and 1 billion years ago, and refuse to leave until the weight of that afternoon has sunk in as deeply as words can carry it. Let us begin again, more slowly than before. You are lying on your stomach in warm, ankle-deep water. The microbial mat beneath you is perhaps 8 to 12 millimeters thick, thick enough to feel substantial, thin enough that you can sense the firmer sediment beneath it. The texture against your skin is complex, slightly slimy from fresh EPS, slightly crunchy from precipitated micrite, slightly grainy from silt and very fine sand trapped during the last high tide. The dominant color is dark olive green grading to black where sulfide-rich layers lie close to the surface, but in direct sun, the topmost filaments glint with purple and reddish-brown pigments, cytomin and mycosporine like amino acids protecting against UV. Breathe in. The air smells faintly of salt, wet stone, and something indefinable, not quite rotten eggs. The H2S stays trapped below the mat except in small seeps. Not quite fresh-cut grass, no lignin, no terrestrial volatiles, but a subtle organic sweetness mixed with mineral sharpness. It is the smell of life that has never learned to fear death in the animal way, because death here is only a change of redox partner. Close your eyes. Listen. There is almost nothing to hear. The tide is near slack. Small wavelets lap every 6 or 7 seconds against the outer edge of the mat, producing a soft, irregular SHHH that never quite synchronizes. Every few seconds, a larger oxygen bubble escapes from somewhere under your chest, a tiny plink as it breaks the surface film. Very far away, perhaps 2 or 3 kilometers across the lagoon, a low-frequency crackle suggests a distant thunderstorm cell forming over the open ocean, but it is too remote to carry thunder. No birds, no insects, no frogs, no fish splashing. No wind making leaves rustle, there are no leaves. The silence is not absolute, but it is biological silence. Everything audible is either water or atmosphere or geology. Now open your eyes and look down at very close range, centimeter scale. The mat's not smooth. It is covered in tiny irregularities, small gas domes where oxygen or methane has collected under the EPS sheath and lifted it like a blister. Networks of filaments crossing each other in random but locally ordered patterns, patches where carbonate precipitation has begun to form a hard crust, white to pale gray, slightly translucent when wet. Occasional clusters of mineral grains, quartz silt, feldspar fragments, clay flakes that have settled during the last slack water and become glued in place. If you could zoom in further to micrometer scale, you would see the filaments themselves. Long chains of cells, each cell perhaps 5–10 μm wide, surrounded by a layered sheath of polysaccharides. Some filaments are straight, others gently curved or coiled. Among them float smaller coccoid cells, unicellular cyanobacteria, or perhaps early eukaryotic phytoplankton that have not yet left unambiguous fossils. Heterotrophic bacteria crawl slowly along the sheaths using type IV pili, scavenging organic exudates. Phages, viruses drift in the thin water film, occasionally attaching to a host cell and injecting their genome. Everywhere there is motion, but none of it is intentional in the animal sense. Motion here is diffusion, convection, Brownian jiggle, gliding motility powered by proton motive force, phototaxis guided by membrane-bound pigments, never flight, never pursuit, never display. Now raise your gaze to horizon scale. The tidal flat stretches away in gentle curves. To your left, seaward, the water deepens gradually to 1/2 meter, then 1 meter, then disappears into a hazy blue-green distance where wavelets sparkle under the low sun. To your right, landward, the mat gives way to a slightly higher, drier zone where desiccation polygons have formed, cracked, curled polygons of mud and microbial remains that will be rewetted on the next spring tide. Beyond that zone lie low, rounded stromatolitic bioherms, clusters of domes and columns rising 20-80 cm above the flat, their flanks stained dark by endolithic communities that bore into the carbonate surface. Farther still, perhaps a kilometer away, a small volcanic island rises. Black basalt flows from some Archean Proterozoic transition eruption, now deeply weathered, colonized only by thin biofilms in cracks and hollows. No trees, no shrubs, no grass, just rock, mat, water, sky. The sun is perhaps 2 hand widths above the horizon. Its light comes in long, low angles, turning every small ripple and every dome into a miniature landscape of light and shadow. The water between the domes acts like thousands of tiny mirrors, reflecting the amber sky and the redder sun. If you stay until sunset, and then through twilight, and then through the first stars appearing, you will witness one complete diurnal cycle. Daytime, maximal oxygen production in the topmost layer, oxygen, bubbles forming and rising, pH rising slightly. As CO2 is consumed, sulfide oxidizing at the oxic-anoxic interface, methanogens in the deepest layers producing CH4 that diffuses upward and escapes nighttime. Oxygen consumption by respiration outpaces production. The entire mat becomes anoxic within minutes. Sulfate reduction dominates fermentation of stored polyglucose. Sulfide accumulates near the surface. Small amounts of H2S diffuse into the overlying water and react with any remaining traces of Fe2 or Mn2 dawn. Light returns. Photosystem II reactivates oxygen production, resumes the cycle, begins again. This cycle has repeated, with only gradual evolution of the participating lineages, approximately 1 trillion times since oxygenic photosynthesis became widespread. Billion years ago, 1 trillion days and nights of essentially the same chemical ballet. Now try to feel that repetition, not as boredom, as endurance, as faithfulness. As a form of devotion that does not require consciousness or intention. Each cyanobacterium that fixes one more CO2 molecule, each sulfate reducer that returns one more sulfur atom to the cycle, each methanogen that releases one more CH4 molecule to the atmosphere, none of them knows it is participating in a planetary respiration system that has kept Earth habitable for billions of years. Yet they do participate, they do keep the system running. And because they keep running it quietly, blindly, tirelessly, the planet accumulates oxygen, oxidizes iron, precipitates manganese, buries carbon, stabilizes climate, prepares seawater chemistry, evolves eukaryotic complexity in the background, invents sex, invents multicellularity, invents developmental patterning, all while never once needing anything that could be called an animal. When the first small bilaterian finally scratches its tentative trail through the mat sometime around 555-541 Ma, it is walking, quite literally, on the accumulated labor of a trillion microbial generations. That is the final image we can carry away. Not a dramatic sunrise over a Cambrian reef, not a trilobite eye reflecting the new world. Just a microbial mat at twilight, still quietly photosynthesizing with the last photons, still quietly respiring with the stored carbohydrates, still quietly cycling the elements that will one day allow bones, shells, eyes, brains, cities, telescopes, poems. Still here, still sufficient, still sovereign. After animals come and after animals go, it will almost certainly still be here, doing the same patient work under the same amber sky or whatever sky remains. That is what Earth was really like before animals, and in the longest perspective, that is still mostly what Earth is like even now. We were only visitors.